Hello and welcome to the Pharma Forum podcast. I'm your host, Jonah Comstock. The early success stories in cell therapy have mostly been CAR T therapies, with T standing for T cells. But T cells aren't the only option for cell therapy. Promising work is being done with natural killer, or NK cells, a different kind of immune system cell that could work even better in some ways. My guest today is Dr. Fred Aslan, CEO of Artiva Biosciences, and he's going to tell us more about this new avenue in cell therapy and some of the work he's been doing. Welcome to the show, Fred.
Thank you. Thanks for having me.
First of all, tell me a little bit about yourself and about Artiva. How did you what's your background and how did you get into this space?
Yeah, so I'm a physician by background. I transitioned over to industry around 20 years ago. I have stints in venture capital, and I've also been an entrepreneur starting 13 years ago. That's when I made the transition over to the executive side. And I've had an opportunity now to work in a number of different companies, both in the medical device space and in the biotech space. And Artiva, we were founded 5 years ago. We're a San Diego-based company. We're exclusively focused on the biology of NK cells. We have approximately 80 employees, with half of them being in our tech ops area, responsible for manufacturing of our NK cells. We've raised over $200 million to date by a great group of venture capital, crossover, and public investors. And we are in the clinic with our lead asset, which we call AlloNK, for allogeneic NK cell.
Is that the one that there was just a press release about today?
Correct. So today we announced that we have dosed the first patient in our trial with AlloNK in autoimmune disease. So as far as we can tell, this is the first patient in the US to be treated with an allogeneic NK cell in the context of autoimmune diseases.
Awesome. So I was at CAR TCR last year in Boston, and I remember hearing a little bit about NK cells from a few companies. Your company may have been among them, but it seems like it's a much narrower kind of space right now in terms of the number of folks who are in it. Tell me a little bit about these cells and what makes them special, what makes them different than and perhaps superior in some ways to T cells.
Sure. So NK cells stand for natural killer cell, as you know. It's a type of immune cell. Just like T cells, they can be highly cytotoxic, right? So our immune system depends on them to kill defective or foreign cells. We are focused on exploring the biology of NK cells, which the main relevant piece of the biology to us is the ADCC mechanism. So ADCC stands for antibody-dependent cell-mediated cytotoxicity. And this is really important for how monoclonal therapies work. So if we take a monoclonal antibody like Rituxan, for example, right, and we think about how does it work in the context of an autoimmune disease, the way it works is the monoclonal, using Rituxan as an example, would bind to CD20, which is an antigen on the surface of a B cell. But then an NK cell binds to the other end of Rituxan, and this construct of Rituxan bound to an NK cell and bound to the B cell activates the NK cell, and it causes the NK cell to kill the B cell. So very similarly to the way CAR Ts work, where the T cell is engineered with a CAR to actually go after the antigen on the cell of interest, in this case, we use a combination of our NK cell together with the monoclonal because it's been very well described in the literature how important NK cells are in the efficacy of monoclonals. So what makes them special is the fact that they do work in this mechanism of action. Today, we have clinical data from a trial that we ran in non-Hodgkin's lymphoma, which shows that this combination of NK cells together with rituximab was able to actually drive some pretty interesting responses in cancer patients and to deplete the B cells in these cancer patients. So we can talk more about that later. But the really big advantage over CAR T cells is that CAR T cells are known to actually lead to some side effects. And these side effects is what keeps most patients requiring hospitalization in order to be administered the therapy. So our vision is to develop an off-the-shelf product, and we can talk about, this difference between having an off-the-shelf off-the-shelf NK cell versus having an autologous T cell construct, and the difference is there. An off-the-shelf product that at the same time has a differentiated safety profile, which would allow it to be used outside of the hospital setting, right? So in an ideal world, we could use cell therapy in a community setting where patients can receive therapy in an infusion center and then go home without requiring the hospitalization. And then the final advantage is that at least we have developed a highly scalable process to manufacture these NK cells, which means that we can drive down the cost of actually manufacturing this therapy. And that would give us pricing flexibility because, as you know, one of the big limitations of auto CAR T today is that the price is actually really high, right? In the order of like $500,000. So part of the reason why auto CAR T is so expensive, it's because it's very expensive to manufacture the product. So one of the advantages of NK cells, particularly utilizing our process, is that we have the ability to scale these NK cells pretty dramatically.
So that's a great overview. I want to take some of those in turn. First off, why can you do this off the shelf? I mean, are these cells kind of universal, that the same NK cells will work in any person? And why would that be the case as opposed to other cells where it's advantageous to use the patient's own cells?
Yeah, so people started testing around 15 years ago this notion of using an allogeneic NK cell, taking an NK cell from someone and then giving it to another patient, right? And what they were able to see is that NK cells are actually fairly well tolerated when you're receiving them. One of the problems of T cells is if you take a T cell from one patient and you give it to another, there's an immune rejection of that T cell. But NK cells are able to actually function well in an allogeneic process. People started initially matching HLA as a way to avoid that immunogenicity. But then they found that even without HLA matching, you can still transfer NK cells from one patient to another, and they're pretty well tolerated. So this notion of manufacturing is that, okay, once you know that you can actually take NK cells from one patient and give it to another, then you're now in a place where you'd almost call that a transplant, right? Like you're asking, I think you asked me a question, how is this not like a transfusion? Because in a transfusion, you actually take cells from one patient and you give it to another. The difference here is that, if you were to strictly take NK cells from one patient and give it to another, you're very constrained in terms of the amount of these procedures that you can do, right? If every time you needed to treat a patient, you needed to actually find a donor in order to take NK cells from them to give to a patient, then, this is not very scalable. For example, we are now trying to go after autoimmune disease where you have tens of thousands, if not hundreds of thousands of patients that may need this. You can imagine that, a therapy is not really going to be very scalable if you need to find tens of thousands of patients to donate blood or NK cells in order for that to happen. So part of the secret sauce in our manufacturing process is that, first of all, we source our NK cells from umbilical cord units, right? And we have many banks across the country and across the world with umbilical cord units that are available. From one umbilical cord unit, just to give you a sense of the scalability of our process, we can make 4,000 vials where each vial has 1 billion NK cells, right? So we're talking about taking one umbilical cord unit and generating enough product to treat hundreds of patients, right? And the way we manufacture this is once we actually scale up the NK cells and we place them in vials, we can cryopreserve them and we can store them for years, right? And then these products can be sent through cold chain logistics to wherever patients are going to be treated. The product is thawed at the bedside and then they're infused into patients. So the big difference between what we're doing in auto CAR T is this concept that we're generating a universal product, off the shelf, meaning that it's available whenever a physician wants to treat a patient.
That's really fascinating. And just to do a little plug, if you're a new listener to the podcast, we did an episode all about birth tissue donation and some of the issues around that. So that worth giving a listen if you're interested in that beginning of that process. But I wanted to ask about the other piece. You said that, the NK cells work in conjunction with another drug or therapy, right? What's that? And I mean, if you're able to sort of scale and manufacture the NK cells at this rate, is that other piece able to keep up too?
Yeah, absolutely. So the other piece is monoclonal antibodies, right? So as you know, when we think about biologic drugs, right, they are generally either protein therapeutics or they're monoclonal antibodies. And monoclonal antibodies are probably the largest category, right? So very high profile drugs ranging from rituximab to Humira to Herceptin, right? These are all monoclonal antibody drugs that are available off the shelf and they are available for treatment. And so the way we utilize these monoclonals is, as I was explaining to you before, the way a CAR T knows where to go to actually do the killing is one engineers in a CAR, right? The CAR allows the T cell to be targeted towards the antigen of interest and the tissue of interest, right? What we do is we use non-genetically modified NK cells, right? And we also use these NK cells in combination with these monoclonals. So what happens is the monoclonal does the targeting, right? You can imagine that you give the monoclonal finds all of the antigens of interest, and then by giving the NK cell, high amounts of exogenous NK cells, we're giving these NK cells an opportunity to now find these monoclonals and to kill those cells of interest. So generally, when you use a monoclonal, you are relying on a patient's endogenous NK cells to actually be doing the killing. But when you talk about late line cancer patients or you talk about patients with autoimmune disease, they don't have the same level of healthy NK cells that healthy individuals do. And so our hypothesis from the beginning has been that this addition of exogenous, large amounts of exogenous active NK cells would enhance the killing of these antibodies. And that's exactly what we are seeing in the clinic.
So you're essentially calling in reinforcements for the patient's immune system.
Yeah, we are in a way enhancing the activity of the monoclonal antibodies by actually providing reinforcement, as you're saying, of the immune system, since the patient's immune system is so critical in driving the efficacy of these monoclonal antibodies.
And these NK cells, they don't have, as far as you can tell so far, the same risks when it comes to side effects and cytokine storms and that sort of thing.
That's correct. Part of the reason for that is because the way the CAR Ts work is they're administered and then they go through a very rapid expansion process, right? Now, as they are expanding and as they are killing cells, they do release cytokines that then ends up revving up the immune system. And that can lead to cytokine release syndrome, as you mentioned. And cytokine release syndrome can be very dangerous, right? So we have learned over the last, 10, 15 years since we've been using CAR T, how to manage this in a hospital setting. Typically, a patient initially starts to develop fever. You try to control that fever. If that fever cannot be controlled, then you can use agents that actually try to reduce the inflammatory milieu. Very specifically, a drug called tocilizumab, which is an anti-IL-6 monoclonal antibody, is used. And when used, that can actually control that cytokine release syndrome escalation and avoid it from getting to a grade 3 and grade 4. By the time you have grade 3 and grade 4, you're having to go to the ICU because you develop a life-threatening condition. But nowadays, if you're hospitalized and the fever starts getting out of control, the use of tocilizumab has helped control that. But again, you're left with a procedure where you're having to be hospitalized and you're having to be watched very closely in order to avoid that CRS from escalating. The advantage of NK cells, and that has been generally true of the entire field of NK cells, is that we don't lead to that level of CRS or that, or that, because CRS is something that happens in the vast majority of patients that are taking CAR T. But because of the use of tocilizumab, we can control that and retain it to grades 1 and 2. When you look at NK cells and the incidence of CRS and how much it escalates, it's actually a very small fraction of what one sees with auto CAR T. And that opens up the opportunity for NK cells to be dosed in the community setting. So what I can tell you is, as part of our NHL trial, we did treat and manage the vast majority of our patients in the outpatient setting. They only had to be hospitalized if they developed a fever that the physician was concerned with, or if they developed a serious infection. But that happened in a minority of patients, right? So our goal is to have this therapy be something that you can do in the community setting.
So, in the news that came out today, one thing that struck me is that we're looking at autoimmune disease. So far, a lot of this CAR T stuff has been around cancer, specifically blood cancer. So, is that a feature here of the NK cells, the different technology? What's the broad number of kind of indications where this might be able to help?
Right. So AutoCAR T was initially developed in cancer, and there was a study that was published in 2022 by a German academic, where he used AutoCAR T in the context of patients with autoimmune disease. And this study really raised a lot of interest in the industry, because the efficacy that this investigator saw by using AutoCAR T in autoimmune patients was actually quite dramatic. Almost, I believe, all of his patients ended up becoming drug-free, disease remission. And that's something that is unprecedented in the treatment of a lot of these autoimmune diseases. And the mechanism is actually fairly similar to the mechanism in cancer. So in cancer, particularly NHL, which is probably the biggest indication where AutoCAR T is approved. It's right now approved in NHL and in multiple myeloma. NHL specifically is a B cell cancer, where the B cell transforms into cancer and it grows. So the mechanism of action of AutoCAR T is to use the targeting feature of the CAR T to bind to an antigen on B cells and to kill all the B cells that are present. That way, when you're doing that, you're also killing the cancer cells. When you're talking about autoimmune disease, you're also talking about a B cell disease. In the case of an autoimmune disease, the B cells are producing autoantibodies that attack the internal organs of a patient, right? So because they are both B cell driven diseases, and because AutoCAR T has been efficacious at actually targeting B cells and killing them, it was actually surprising and very encouraging that that same mechanism could be applied in autoimmunity. So if you are an NK cell therapy company like we are, you observe these results and you say, well, we are already seeing a deep degree of B cell depletion in cancer patients. We're seeing that using our therapy, we're able to drive responses in NHL patients. So the logical conclusion is to also try this in the context of autoimmune disease, where again, you're trying to deplete B cells, and so that you can actually improve how the patient is doing. Very important in that study, what the investigator, Dr. Schett, was able to show is that by killing all of the B cells during a certain period of time, you cause what's called an immune reset, where the B cells reconstitute after that. And when they reconstitute with what's called a naive phenotype, i.e. a healthy phenotype that stops producing the autoantibodies, at least for a period of time, and avoids patients getting the symptoms again. So that's why we have also expanded our efforts to go into autoimmunity, because we believe that we could pursue the same MOA to potentially drive efficacy in these patients, but with a differentiated safety profile than AutoCAR T. Again, AutoCAR T today is relegated to a hospital setting, whereas in our case, this is an off-the-shelf product that is available whenever a physician deems that they want to enroll a patient in the trial.
It is really fascinating. I mean, that the various different benefits here, but that notion that it could be so much cheaper to produce, and therefore cheaper to the physician and to the patient, really seems like it could be a game changer. This could kind of take over as a primary treatment. I mean, what's your vision for? I know you're in the early phases here, for where you go from here.
Yeah, the vision, and this is shared amongst all of us in the field, is that if we are able to, in clinical trials, to demonstrate a degree of efficacy that we're hoping for, then this could really unleash the number of patients that can actually access this therapy. Because you have to understand, like, you probably know what the auto CAR T process is like, but just to walk you through that, because that'll explain why a procedure like that becomes very challenging to be used and available for tens of thousands, potentially hundreds of thousands of patients. So the way it works is you screen a patient to figure out if they're eligible for the therapy and if they meet all of the inclusion exclusion criteria. If they do, then you have to actually bring them in and you have to do what's called apheresis. During an apheresis procedure, you're sitting down in a clinic and blood is removed from you during probably a 3 to 4 hour procedure where they're trying to withdraw T cells, right? So that then those can be engineered on. So you complete the apheresis, you go home. Now you have to wait several weeks for the product to be manufactured for you. So this is going to be customized for you. They take your cells, they engineer in the CAR, and they engineer in the things that they need to do, and then they expand those cells. And then several weeks later, you get hospitalized so that you can receive the therapy, and you remain hospitalized for a 10 to 14-day period to make sure that you're not going to have any acute effects. And then even following treatment, you have to stay relatively close to the hospital. You have to be within an hour drive of the hospital so that if for whatever reason it's determined that you have a risk for CRS, you can be quickly admitted and be treated with. So when you think about the idea of now tens of thousands or hundreds of thousands of patients having to go through that, that becomes a challenge on a number of fronts. First of all, you need to have enough manufacturing capacity out there in order to be able to produce enough product to treat all of these patients. And we already have capacity constraints today in cancer alone, right? And cancer has, an order of magnitude less patients than autoimmune disease, which is a lot more prevalent, right? So there's, first of all, this notion of manufacturing constraint. The second constraint is because this is largely a hospital-based procedure, you need to have enough hospital beds and the availability of enough hospitals that have the expertise to be able to manage all the steps of this process, all the way from the apheresis to the treating the patients and then managing them, right? Now, you compare that with how biologics are used today, monoclonal antibodies. It's much simpler. With a monoclonal antibody, a physician will be practicing in the community setting. They will have an infusion center either in their clinic or just around the corner in the community setting. And if they determine that one of their patients need to be treated, they pretty much just schedule a time for the patient to go into the infusion center, and then the monoclonal antibody is given, right? It is an extremely scalable process, and which is why these monoclonal antibodies have been so effective and accessible for people. So what we're trying to do with NK cells, and again, this is true of the majority of everyone in the field, is to try to have this cell therapy be as close to being as accessible as a monoclonal antibody is so that more patients can benefit from it.
Just sort of as a point of curiosity, if these cells are sort of so much better in these specific ways than T cells, and they don't need to be modified, in a sense, you're doing less work on them. Why did the space develop in the order that it did? Why didn't we get NK therapies in the first place?
I have been in the field for only 4 years, right? So I can't really speak to all of the originating work. There's other people that have been following this for a lot longer than I have. The idea behind the auto CAR T is that you have to worry about two things in general when you're using cell therapy. The first one is that the therapy actually persists in the body after you give it to them, because our immune system is known to attack, right? Any foreign cells that actually come in. Even our NK cells, I mean, they generally last for around a week, right? And so the way we give our treatment is we usually give them in weekly doses because, again, they generally last for a week. The idea behind an autologous cell is that that is your own cell that you're genetically modifying. And so therefore, the likelihood that it can persist in the body is a lot better than when you're using an allogeneic product. So it makes sense to me why one would have started with an autologous cell. The other thing I'll tell you, this idea of engineering a T cell so that it can target a particular cell type that you're interested in makes a lot of sense because T cells can be very cytotoxic. And as I mentioned before, because these T cells actually expand once you give them to people on their own, you don't actually need very high numbers of cells in order to achieve your therapeutic effect. When we're talking about NK cells, first of all, they don't have this ability to actually massively expand. So you actually have to give very high doses of NK cells. And that was probably limitation number one, is that it takes, and it took a long time for the field to actually develop massively scalable ways to, generate very high numbers of NK cells that then could be given to patients. And the second aspect, there are two ways to think about this. One way is to add a monoclonal like we're doing, or like others have done, one can engineer NK cells so it can also have a CAR and it can try to do that activity on its own. So to me, it feels like auto CAR T felt like a more linear and more clear approach to try to generate both persistency so it could work as well as the efficacy. And NK cells feel like a cell type that had been studied also for a long time. But once auto CAR T started to work, then people in earnest started to invest more in NK cells to see whether it would work as a suitable alternative with the advantages that I mentioned. And on the dosing, just to give you a sense of order of magnitude, today you can administer around 60 million auto CAR T cells, and those 60 million auto CAR T cells will expand by 100 times plus inside a patient, right? So you can imagine that the 60 million times 100 is generating many more, right? It's like over 1 billion cells that are now active in that patient trying to kill. With our patients, we're giving billions of cells when we give them our NK cells. So this again brings up this notion that NK cells can only be viable if you have a manufacturing process that allows you to produce very large numbers of NK cells so that they can be available off the shelf and given to patients.
So that's really one of the big innovations you guys are bringing is just this manufacturing logistics of actually being able to do this because you're able to create a large enough supply.
That's correct. So we are using technology from a Korean company, GC Cell. GC Cell was actually the first company, as far as we know, to actually give an allogeneic NK cell to a patient. I think this was back in 2006. And since then, GC Cell has been developing a process to scale and expand NK cells. So the process that we use today has had the benefit of over 15 years of work that GC Cell has put into this process. So the way our collaboration with GC Cell works is we own the ex-Asian rights to the technology, to manufacturing and all the products that emerge from this platform of NK cell expansion, and they retained Asian rights to the technology. So it's thanks to their innovative work and then all of the improvements that together we have built on them that today we are where we are, with such a mature process.
Not to push you too far towards the whatever's proprietary here, but why cord blood or umbilical cords is a place to start? I mean, adults produce NK cells too. What is it about that makes it kind of the best starting point?
Yeah, I would say that there are three main sources of NK cells that people have been exploring. One is umbilical cord units, like we are. The second is peripheral blood. And then the third, you're probably familiar with companies that work on iPSC-derived NK cells, right? With iPSC-derived NK cells, you're starting with a stem cell, but then you're helping that cell mature to become an NK cell, right? So you're not starting with a mature NK cell in that case, right? You're starting with a stem cell. If we compare and contrast umbilical cord units with peripheral blood, and GC has had experience working with both sources, what they found is by the time you become an adult, your NK cells have matured, and there are big differences from individual to individual. So if you are extracting NK cells from an individual, there's a very good chance that you will find donor to donor differences across these individuals. That's what GC found when they were developing a process utilizing peripheral blood. With umbilical cord units, what we find is you start off with a much less mature NK cell, but as part of our manufacturing process, which we can talk a little bit more about, this immature NK cell matures in a very uniform and consistent way. So when you look at the final drug product, what we see are very high levels and consistent levels of all the receptors that you would want to see on the surface of the NK cell. And that gives us the ability to have a manufacturing process where the ending material looks very similar batch to batch, regardless of the donor that you're using. Right. So we do know that there's a number of different companies that are innovating, the ability to scale peripheral blood. We're just not as close to the improvements that they have done. But one of the reasons we like umbilical cord units is because of this consistency in the manufacturing process and in the end product.
Is there anything we haven't talked about yet that you really want to discuss, either related to what you do specifically or just general thoughts about the industry you're in?
Yeah, I mean, this is a very exciting time. I don't know how much people appreciate, but every few years there is a groundbreaking study that all of a sudden everybody stares at and they're like, wow, this is such a strong signal. We have to, as an industry, invest the resources to see if we can exploit it. And that's how I think about that German academic study. This notion that you're taking very late stage autoimmune patients and you're able to actually drive them to, drug-free disease remission is really remarkable. So I believe that this is going to be a signal that people are going to be following and exploring for the next decades to come, right? Because we don't have a really good solution for autoimmunity patients. We are particularly excited to have our angle in this exploration because of the advantages of the NK cells that we talked about. But there are a lot of companies that are entering this field now. And this is a really good thing for patients because there are multiple modalities that are being exploited. These multiple modalities are being exploited across a number of different autoimmune diseases. And so that's really exciting. And maybe this is the final point about the breadth of therapies like this. Because this notion that autoimmune diseases are caused by B cells that produce these autoantibodies that are bad for you, this is true of a variety of very big autoimmune diseases, ranging from rheumatoid arthritis to multiple sclerosis to lupus. I mean, these are diseases with prevalences in the hundreds of thousands, right? So this signal that was uncovered in this academic study, pointing to the possibility that you can drive these patients to what looks like a at least near-term cure is actually quite exciting.
Yeah, definitely. And I remember at CAR-TCR last year, I heard a few folks talk about this paper and talk about sort of lupus specifically, but autoimmune generally as like the next frontier. And the other thing I heard a lot of conversation about was auto versus allo and what is it going to take to move us into an off-the-shelf space increasingly. So this really aligns with a lot of what I've been hearing, at that conference, and as I talk to other people about where people are excited about when it comes to the future of cell therapy.
Good stuff for patients. We're very excited to be a part of it.
Well, thank you so much, Fred. It's really been great hearing about what you do. It certainly expanded my horizons a little bit when it comes to cell therapy and what we can do. Good luck with everything, and I look forward to seeing some of the results from some of these trials as they progress further.
Very good. Thank you. It was a pleasure speaking with you.
That concludes this episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about other installments in the series at pharmaphorum.com/podcast. The pharmaphorum podcast is also available on iTunes, Spotify, Acast, Stitcher, and Podme, where you can find and subscribe by searching for pharmaphorum. And don't forget to visit our website, where you can sign up for daily news and analysis bulletins, and to follow us on Twitter at pharmaphorum. Thanks for listening.